Hello, everyone. This is Matt Ricker at North Carolina State University, an assistant professor of pedology and land use. I'm going to talk to you today about some of the research that we're doing in the Coastal Zone Soil Survey avenues here in North Carolina. And I'd like to acknowledge my co-authors, Greg Taylor with the USDA NRCS and the Coastal Zone Soil Survey team, and then Lori Gorczynski, who's a graduate student here at North Carolina State University. So today I'm going to talk about some ways that we're pairing our subaerial and subaqueous evaluations of soil to understand impacts of sea level rise on the coast on histosol landscapes. And so traditionally we've ended our evaluation of soils here at the shoreline, and we're pushing our soil maps and our soil interpretations out into the subaqueous environments, and we're starting that here in North Carolina. I would like to highlight that there are additional resources at the Soil Science Society of North Carolina virtual meeting this year. There are two posters that are associated with the Coastal Zone projects. One is from Lori Gorczynski. She'll be talking about histosol blue carbon stocks and some of her specific work for her graduate degree. And then we also have Sarah Jordan, who will be talking about using GIS to evaluate both soil systems and on-site septic systems and their vulnerability in North Carolina to sea level rise. So why coastal soils in North Carolina? Why is this a big deal? I moved in 2018 to North Carolina State University. I moved from Pennsylvania. I've been doing research, environmental research on coal mining impacts in Pennsylvania, which is a big deal there. In North Carolina, not so much, and so I needed a way that we could apply pedology consistently and that would be important to the citizens of North Carolina. And during my initial meetings here at NC State, I had multiple meetings, and one of the top priorities was increasing coastal resilience. And so coastal resilience is helping communities on the coast adapt to predicted issues with climate change, specifically sea level rise, and then associated issues, flooding, salinization of soils, impacts to urban systems, engineering systems, environmental interpretations, all kinds of reasons that we're worried about the coastal zone here in North Carolina. So why is this important here? According to NOAA, we have the 6th longest tidal coastline in the US here in North Carolina, and we have 1.2 million residents that live in our coastal counties, and many millions more visit per year. And the big thing here is our current soils information for our coastal zone soils is severely lacking. We have very little information both in the subaerial and subaqueous environments in North Carolina. And so we've begun to work with the Coastal Zone Soil Survey team through the USDA NRCS to really increase this information that we have. And so this summer we were doing subaerial and subaqueous paired site analyses. And so here we're on land, so we're in the subaerial environment, we're under air. And then offshore we have a coring pontoon boat that's looking at the subaqueous soils. And so we're working hand in hand with USDA to really make these connections. I'm going to talk a lot about blue carbon today. If you haven't heard the term, blue carbon is essentially just carbon that's associated with oceans and coastal areas that are influenced by tidal processes. So this carbon can be locked up in soils, and it can also be in the vegetation. The soil system plays a central role in any carbon cycle, so the blue carbon cycle is no different. And it's traditionally understudied. So currently we have very little information about our subaqueous or our semi-diurnally flooded areas like these tidal flats or these areas of the estuary that are underwater. And then we have, conversely, very little information about these areas as well that are histosols or wetland soils that are associated with marshes, and then down here are tidal swamps, because these are poor agricultural areas for a number of reasons. They're very wet. And then two, they have very high salt content on the coast. And so they're very poor for agriculture. And originally when the soil maps were being created in these areas in the 1980s, these were non-priority areas. And we currently have no subaqueous inventory whatsoever in North Carolina in our tidal areas. So we really need some baselines to understand how much carbon, blue carbon, is stored on the landscape and how that's changing as salinization and flooding is increasing. How's that changing as sea level rise is occurring and submerging these subaerial environments, and they're becoming subaqueous soils. One of the major things since I got into thinking about blue carbon is what's the boundary on the landscape between blue and terrestrial carbon? And there's no real hard and fast rules here. Some folks believe that blue carbon should only be the carbon associated with marsh systems, so systems like up here. Others like me, because the Pamlico and Albemarle sounds are so extensive, you're able to travel in some cases 70, 80 miles from the marshes, and you still have tidal influence or microtidal influence, as well as influence of storm surge events that bring in salt to these areas periodically. And so I tend to think that we should think of blue carbon as anything within the estuary that's having any tidal influence with salinity. And so that would include our tidal fresh systems that have woody vegetation like the swamp, and our marsh systems as well. But this is up for debate. So just to let you know a little bit about our major study area. So the Albemarle Pamlico Sound is obviously in northeast North Carolina. Albemarle Sound is to the north, the Pamlico is to the south. This is a pretty nice map that shows both the bathymetry, or the underwater components of the system, as well as the elevation, so the components of topography up on the landscape. So both our subaerial and our subaqueous environments. And the thing that makes this area unique is that it's the second largest estuary in the US, behind the Chesapeake Bay, but it's the largest lagoonal system in the United States. And so what a lagoon is a system that's cut off primarily from lunar tides. And so this is a very large water system. It's over 7.8 or 7,800 kilometers squared. It's a very large system. And lagoons are cut off from lunar tides, so they don't receive the semi-diurnal or twice daily flooding that's predictable in most areas, because the outer banks interfere with the tidal range in this area, and they cut off the lunar tides effectively. So these are called microtidal areas that are influenced more by wind than they are by actual lunar tides. And the way that salt and tidal influence gets actually into the Pamlico and Albemarle is through the inlets. So we have three inlets: the Oregon Inlet up here, we have the Hatteras Inlet, and then the Ocracoke Inlet here. This is considered a wind-driven system. Sometimes this is called seiche, which is basically blowing. Think of it as a bathtub with salty water that's coming from the ocean, and then that's mixing with fresh water that's coming down our river systems. And basically we have all kinds of interesting salinity gradients that are occurring. The major thing that's interesting here is that we have very shallow depths, on average 2 to 3 meters, so 6 to 10 feet. You can see that here. So in our subaqueous evaluations of soils, we can pretty much look at this as a continuous subaqueous environment that we could actually map from a soil survey standpoint. This is considered a drowned coastline since the last glaciation. So during the last glaciation, which was at its maximum about 20,000 years ago, Laurentide ice sheet, which during the Wisconsin glaciation would have extended down to northern New Jersey on the Atlantic coast. And then ever since that point, it's been melting back. And as it's been melting back, sea level has risen significantly. So during the last glaciation, with all that water tied up on the polar ice caps, sea level would have been out here to the edge of the continental shelf, about 60 miles from its current configuration. And over the last 20,000 years, sea level has risen such that the coastal outer banks have moved in 60 miles inshore. Also going with glaciation, you might not think glaciers are important in North Carolina because they never got here. But as a glacier came down during the last glaciation, it depressed basically the soft mantle underneath the crust, and it created sort of a teeter-totter effect. And what happened is basically by pushing down, once you remove the weight of the ice that used to be there, the land is basically rebounding. So it's coming up in certain areas that had ice on it, but then other areas that were basically pushed up in front of the ice are now sinking to come into equilibrium, and we're in one of those areas. So this area of North Carolina has significant what we call isostatic rebound. So the land is subsiding and sea level is coming up. And what this leads to is the highest relative rates of sea level rise in North Carolina in this region. It's about 2 times the global average of relative sea level rise. And the predictions from NOAA are about 1 foot of sea level rise by 2050 and about 90 centimeters or 3 feet by 2100. Why is this an important issue in North Carolina? Well, notice this is the elevation. It's kind of this light sort of green and then this light blue. This is from 0 to 5 feet above sea level. So 3 feet of sea level rise is going to be very impactful in this region. And just for your reference, if we melted all the ice on both Antarctica and Greenland, that would be enough sea level rise to basically fill the coastal plain to the fall line here. So we have a lot of area in North Carolina that is susceptible to sea level rise, and we need to take this seriously and think about how soil science can be used for adaptation and mitigation strategies on the coast. So what does this look like, this projected 1 foot and 3 foot sea level rise? We can look at the NOAA sea level rise viewer for this. So this is the Albemarle Pamlico Sound. This is the current configuration of our shoreline, and we can project forward. This is by 2050 with 1 foot of relative sea level rise, and you see all these light blues that are coming in. These are areas that are predicted to be inundated with shallow water by 2050, and really where it becomes a problem is by 2100. If we get 3 feet of relative sea level rise in this region, this is what the landscape will look like. This is the Alligator River, will be completely cut off from the Outer Banks. We'll have some islands sitting out here, massive, massive swaths of area in the Pamlico Peninsula that are predicted to be, inundated with a 3 foot relative sea level rise. So this is extremely important. There's all kinds of farms here. There's all kinds of structures and houses. We need to understand what is happening to soils as they're becoming submerged in this ongoing process of sea level rise. So why are we looking at histosols, you might think? Mineral soils, especially in places like the Piedmont where NC State are, dominate the landscape. But as you get to the coast, especially in North Carolina where we have a very high proportion of histosols or organic soils. So we wanted to make sure that when we're starting to look at the coastal zone, we're looking at the soils that are most represented on the landscape first, and then moving to the more rare ones. And so what we did is just query the soil survey data from Surgo. We took it into GIS and we buffered the current shoreline, tidal shoreline from NOAA in GIS by 500 meters. So anything within about 5 football fields of the tidal coastline in North Carolina was pulled from this database. And this is a conservative estimate, as you saw on the last slide. There's way more area that's going to be affected by sea level rise over the next century or so. But when we did this to get a conservative estimate of what the proportion of different soil orders are on the coast, this is what we came up with. This is mapped area in hectares. What you can see is histosols make up the majority of area that's within 500 meters of the tidal coastline in North Carolina, 32%. Second to that is ultisols, then entisols. We do have insectisols, alphasols, and spotisols as well, mapped along the coastline. So our first cut in what we wanted to look at in North Carolina is our most dominating soil within the coastal zone, and that is histosols in this region of North Carolina. This is actually pretty rare for the East Coast. North Carolina has a very high proportion of organic soils that are susceptible to salinization and submersion and erosion due to sea level rise. And also they're organic soils, so they tend to store a lot of carbon. We looked at the data a little further from Surgo. These are the mapped types, or the classification of the types of soils that can be mapped along the coast, or are mapped currently along our coastline. And you see 77% of the histosols mapped are typic haplosaprous. And if you don't like soil classification, all this means is a deep histosol. We have pteric haplosaprous and typic haplosaprous. So these are regular old, highly decomposed, mostly sapric material, OA horizons, histosols. Typic are deep, pteric are shallow. And what is shallow and deep when you're talking about histosols? A shallow histosol is 130 centimeters or less of organic material before you hit the mineral material underneath. A deep histosol is 130 plus centimeters of organics before you hit mineral. So that's sort of the break between a typic and a pteric histosol. If we break this down even further into our pteric and our typic groups, we can see that we have a number of series, but they're just mapping shallow and deep soils. The blue here are freshwater histosols. Our orange are brackish histosols, which is the Currituck and Habani. And then red are more estuarine, and brackish are more saltwater systems. That's the Delaware and Long Shoal. So these are sol series that are mapped on our coastline. And you'll see here that Dorovan is like sort of the utility knife. It's mapped everywhere. That's probably not appropriate. We need way more brackish and estuarine series to really describe what's on the landscape. And you'll see that as sort of we move forward. And I encourage you to look at Lori Gorczynski's work, where she's been looking at, we likely have a need for at least six to eight more sol series just to map what's on the landscape associated even just with histosols in North Carolina. So we really are handcuffed. We don't have a lot of diversity in the types and classification of histosols that are currently mapped, and it gives us very little information for planning purposes for the future. One of the issues we ran into immediately is if we're thinking about remapping histosols and other coastal zone soils, because these were non-priority areas, they're large units that are not broken up by environmental interpretations at all. If we really want to start looking at these units from an environmental standpoint, we need to identify where these new series would exist on the landscape. And the big thing, or the big driver of that, is salinity of the water. So if you have more saline water, we know that leads to differences in the ecosystem. We tend to have more forested systems where it's more fresh water, and we tend to have more marsh systems where it's more salty water. And then there's sort of this gradient in between where we have dying trees that are moving into marsh, but they're not quite there, and we call those ghost forests. And so the first thing we had to do is look at, well, there's a lot of information available on salinity, but it goes back to the 1970s. And so a lot of the salinity maps that are currently available have data from the 1970s all the way up to present that are going to make those interpretations on the landscape, or on the, in this case, in the water, where salinity gradients exist. But that's not really good for current mapping, so we need more recent information. So we took the last 10 years, 2009 to 2019, and each one of these little green dots here is a data point where salinity was measured within the last decade or so. And then we geointerpolated in GIS these salinity gradients. And so the dark colors are more saline, dark blues, and the lighter colors are more fresh. And so what you can see here is we have a lot of river systems that are entering the Albemarle to the north. We have the Chewan River, the Roanoke River, the Alligator River, and then various smaller rivers, including the North River from the north here. And because we have a lot of fresh water, and remember we talked that salinity from the ocean is only allowed into the Pamlico Albemarle through the inlets. The closest inlet to the Albemarle is here at the Oregon Inlet, so it's slightly south. So there's no direct way that salt can come from the ocean into the Albemarle, and because of that, the Albemarle is, in general, not as saline, doesn't have as high average salinity as the Pamlico to the south. So they're completely different systems. So out here we have polyhaline. This is defined as greater than 18 parts per thousand salinity, which is the back of the Outer Banks. And then coming in and almost touching land here near Engelhard. Most of the Pamlico system is this sort of darker blue, but not as dark as the polyhaline. This is called mesohaline salinity range. This is mesohaline is 5 to 18 parts per thousand. And then up here in the Albemarle, we have this break where we go to oligohaline, which is 0.5 to 5 parts per thousand salinity. And then you see this sort of really light colored blue, which is up in the Roanoke and Chewan, and then a little bit here in the Tar River and the Neuse. That's your actual tidal fresh or microtidal fresh area, which is defined as less than 0.5 parts per thousand salinity. So this is extremely important because you see in red and sort of purple here along the landscape, those are histosols that are mapped directly adjacent to water of different salinities, but you get a Dorovan sol series that's mapped all the way up here, and you have Dorovan that's mapped all the way down here, and they have completely different water salinities directly next to them, and that water salinity changes the ecosystem. So are those the same soils? Probably not. So we need to develop a framework to understand what's happening as not only sea level is rising and salinity is increasing, but also as these histosols are eroding and going and being submerged underwater. So it's a complex system. So in order to start evaluating this complex system, we needed to identify some sites that were stratified by ecosystem type and major salinity. And the major ecosystem types we have are what we call tidal forested environments, where the trees are still intact. Then we have ghost forests, where the trees are dead or dying and the marsh is taking over, but neither system is completely established. So it's a transitional system, the ghost forest. And then we have marshes, which would be further out in the sound, closer to the ocean. So as you move from the more fresh systems, which are associated with more fresh water coming to the estuary, you get higher and higher salinities, which kill the trees. And then finally, once you're out here, only our marsh system can persist. So these were situated on a salinity gradient that existed from tidal fresh systems all the way to mesohaline systems. So we call this a longitudinal study. It's up and down the estuary. We had 12 representative sites that all had mapped histosols. Our tidal forest had 0.16 to 1.6 parts per thousand average water salinity. The ghost forest was 3.4 to 8.3 average parts per thousand salinity. And the marshes are 12 to 15.5 average water salinity. Because of COVID restrictions, we didn't have overnight stays. And if you've ever driven from Raleigh down to, say, the Long Shoal River, down and back in one day, just driving is about 7 hours. So it was very challenging this summer, but me, my technician Ruben Wilson, and Lori Gorczynski did a lot of the sampling in the subaerial environments with these conditions. So it was 12 to 16-hour days to get this research done. The methods we use, and we paired this with the USDA NRCS initiatives for the subaqueous. So while we were working in the subaerial environment in these histosols, which is in green here, the NRCS was there with us most of the days collecting cores offshore. And this is very powerful for understanding what is happening as these subaerial histosols are being eroded and submerged with sea level rise. And so generally our methods were, we drove a boat to these sites, we parked our boat on the shoreline, and we basically established a base camp. We cut a transect through the marsh or the ghost forest or the forest, and 20 meters in, we did a full evaluation to 2 meters, 200 centimeters, or 6.5 feet of the histosol. So we did a full description at the 20-meter point, the start here. We collected lab data from the horizons for a suite of information, as well as bulk density samples. And then while in the field the same day, we collected electrical conductivity and pH measurements at this point. At every one of these points, including the one where we did the full description, and these black sort of dots, and the base camp, we measured peat thickness 5 times. So each site, we have at least 25 measurements of peat thickness along these transects. So we understand a little bit about spatial variabilities, the peat thicker or thinner as you go towards the shore or away from the shore in the subaerial environment. And then in this whole 20 by 20 meter square that we created, we use that as a belt transect for vegetation. So we established the corners, and then we looked at the vegetation, what was the dominant vegetation there, including how many live versus dead trees were there. While this was all going on, the subaqueous crew was out on a pontoon boat that's specifically made for coring, and they were coring in the near shore and the offshore environments. So we had 2 to 3 subaqueous cores and full descriptions per site. And the cool thing, as I'll show in the following slide, is we have the old shorelines. So from Soil Survey, they haven't been updated yet. This is actually important because the old shorelines from the 1980s, that's exactly where the subaerial histosol was in the 1980s. We can actually calculate rates of erosion and submersion. We can also understand when we core in here how old or how long since that point was submerged. So it gives us a component of time. So we have space and we have time here. Eventually, we're going to clean up the units and move the shoreline units. But right now, it's actually important to understand, is this nearshore environment different from the offshore environment, which has been submerged for significantly longer in time. So what does this look like on soil map? This is from SoilWeb. So just sort of looking at this in relation to a soil map, our subaerial histosol evaluation, this is Habani soil series, very commonly mapped. This is Dare County. So Dare County was mapped in 1987, or the map was completed, which is 33 years ago. The average distance of this unit, this Habani unit, that the current shoreline has moved from the mapped shoreline in 1987 is about 125 meters. So over 33 years, the average of this Habani unit here has moved inland and been submerged 125 meters from where it was mapped. And if you do a quick calculation, that's about 3.8 meters per year of submergence. So every year we're losing 3.8 on average, or the overall net average, meters of subaerial environment is being submerged. This is quite common in our mesohaline environments. This is right across from the Oregon Inlet, which is here to the east. This is the Habani series. You can see it has a little A. This is actually very common. You get storm surge events that move sand up over top of the organics, but it's not thick enough to count as a buried soil. And so our classification is of the underlying OA horizons. And you can see here, this is a typic haplosaprist. It's deep organics, so it exceeds 130 centimeters. And it's a haplosaprist, mostly sapric material OAs. So we did our evaluation here, and then NRCS did their nearshore and offshore evaluations here and here. And I'll show some data of why this is important in thinking about budgets of carbon, or how much carbon we're losing over time as the ocean has basically pushed from here to here over the last 30 years. And then eventually, we want to clean this up. We want this break to match the shoreline, and then out here, instead of having a W, we would have some symbology in some series that for the subaqueous environment. We're not close to that, but hopefully in the next 10 years or so, you'll actually have units. So when you step off into the water, we have soil units in the subaqueous environment. So it sounds easy on paper, right? We're just going 20 meters in to the landscape. It's, middle of the summer, 91 to 95 degrees. And you have to hack your way through some of the most ominous vegetation in North Carolina. So this is 14-foot high Phragmites. This is black needle rush. It'll stab right through your gloves. We also had a lot of sawgrass, which will cut you like a razor. By the end of the day, this is Reuben Wilson. You look like Reuben here. Looks like you've been changing oil. We are looking at Histosols, so we're using Macaulay augers primarily in the subaerial environment. There's no other way. This is like fluid material. You can't really sample it. So this is your organics transitioning, in this case, into our more fluid mineral. And so this is an intact half core from our Macaulay auger. You can see here that I'm doing basic chemistry actually in the field. These systems, you have to measure pH and electrical conductivity very quickly, because once you remove the soil from the actual intact soil system, you expose it to oxygen. A lot of chemical reactions, especially related to reduced sulfides, occur, and you have rapid changes in pH and soil chemistry. So we do most of our initial soil chemistry right in the field, and then we immediately put everything on ice and then freeze it until we do further analyses. So it's extra precautions that are needed when you have sulfides in the system. So it looks easy on paper. It was brutally hard to do, and that's probably why a lot of people haven't gone in these areas. We did see a couple cottonmouths. We did see a black bear as well. No gators, so it was a pretty good summer. So for those of you that have never seen a subaqueous soil core, this is a video of a subaqueous soil core being taken. This is a very long core, I believe like 30 meters or 9 feet. This is in the Alligator River Ghost Forest site. You see the dead trees here. Just something about this, it basically vibrates at this collar. It vibrates this aluminum pipe. The pipe then vibrates and essentially sonicates the soil and makes it liquid, and then you can just push, almost like butter, you can push the tube down. This is called vibrocoring. It's one of the most viable ways to get intact cores from subaqueous environments or underwater environments. So this is a nearshore environment that's just been submerged recently within the last few years. And then the boat would move further offshore and do this again. For each one of these sites, the NRCS took 3 replicate cores within the moon pool. You can ask Greg Taylor at some point. These are really highly customized pontoon boats that are specifically made for this purpose. So it's pretty exciting that we have one designated here in North Carolina for these subaqueous evaluations. And I think that's all I'm going to say. So it's a pretty loud process. Obviously, you have to have your PPE and mask on and things like that. But this is us taking a really long vibrocore. So that's the process of getting the tube down into the subaqueous environment. Then we use winches on the boom here to pull it out. Then it's capped. Then it's stored in a refrigerator and then split, and we can make a description. When you split these tubes, this is what you get. This is the examples of our nearshore environment versus our offshore environments. Pretty much we saw this for all of our sites that are nearshore environment. When you're associated with submerged histosols that are on land, what you get is a WAS-ist. So WAS is the classification. WAS is German for water. Ists, so these are underwater histosols. And they're defined as having more than 40 centimeters of organics. So in the nearshore environment, we have, in this case, like a meter or so of organics, the dark colors here, over the mineral underneath. So these are WAS-ist, underwater histosols. Every time that we moved offshore, once you move beyond, let's say 75 meters away from the active shoreline, you would get what we call WAS-ents, so underwater entosols. You can see here there's just this little tiny A horizon on the surface, and then it's just stratified sands and clays. Cool nonetheless, some pretty nice colors, some abrupt boundaries, some interesting redox here, all kinds of interesting features that we're seeing in these subaqueous evaluations. I forgot to mention that video that I just played, that was the first subaqueous evaluations in North Carolina in the Alligator River. So at least from a tidal standpoint, there'd been a little bit of work done in Lake Mattamuskeet previous to this. This is the first tidal subaqueous core. So it's fairly exciting that we have this data now. So just like any soil, we do full descriptions of all the different horizons. We take samples. Those have been shipped off to Lincoln, Nebraska for the Kellogg lab to evaluate, but because of COVID-19, all of our laboratory work has been way behind. So I'll talk a little bit about some of our initial findings, but not too in-depth about data because we just honestly don't have it back yet to report. So just something about how much we got done. It was a lot. So subaerial on land histosols, we evaluated 13 different pedons. All sites were complete. We sampled 83 horizons, 61 were organic, 22 were mineral, 9 were A horizons, 13 were C horizons of different types. Peat thickness in the subaerial or on land environment was measured 105 times. We have incubation pH. This tells us for classification if there's sulfur or sulfides within the column. And then total carbon nitrogen on all samples has been submitted. We do have data on this, but we're pending a lot of quality control on that. So that data will be presented this year at later meetings, but if you're interested in that, shoot me an email. The subaqueous histosols and entosols are WAS-ists and our WAS-ents. Most sites have been completed. There's a couple sites that need to, just because of logistical issues, be revisited, but 16 pedons have been fully described, more are pending. Currently, we have 95 horizons sampled, 23 are organic, 72 are mineral, 17 of those are A horizons, 55 are C horizons. All this is pending, so we'll have basically equal information, and this is a very important study. It's one of the first to look at what's happening as the shoreline is being submerged, specifically with histosols or organic soils. Our preliminary data, you can talk to Lori Gorczynski or check out her poster. She has a lot more information on this, but this is right off of her poster, but this is preliminary peat thickness data, so that 105 times, so we measured how thick the organics were over the mineral. And I mentioned we have fresh ghost forest and marsh sites, and what we're seeing is significant differences in peat thickness as you transition from the fresh site. Saltwater intrudes and kills the trees. The trees completely die off, open the canopy, and then the marsh takes over. And what we see is, in the subaerial environment at least, this is all on land, that the peat thickness varies significantly by ecosystem type. It's 239 centimeters on average thick in our fresh system, and then it is significantly less in our ghost forest, 129 centimeters, and then it's significantly more than the ghost forest, but still less than the freshwater system in the marsh, 171 on average centimeters. The most variability that we're seeing is in the ghost forest. And what this is telling us is that there's something going on as saltwater is intruding into these healthy freshwater histosol systems that we're losing peat. And so we're getting a short-term loss in the ghost forest. And I encourage you to look at Lori's poster because there's a number of reasons that this could be. Lower net primary production. This is plant biomass produced per year that goes into the soil and becomes peat, right? So very high, typically in other studies that have measured this, we have very high net primary production on the end members. The freshwater site has high net primary production. The marsh has very high net primary production. And both of these have very stable root systems. In between, as that transition's occurring, you have dieback of the trees, the roots are dying back, the marsh isn't completely established. It really, you're not getting as much carbon coming in from the plant production, but also you're getting sulfate that's coming in with the oceanic water. That sulfate can be used as an alternate electron acceptor, which means that the microbes can break down organic matter a little more efficient in these systems. And the big thing that I think is happening, and you see it all over our coastline, we're getting more physical erosion. This is a ghost forest site in the Albemarle Sound. And the old shoreline was like hundreds of meters out 30 years ago. Once those trees are dead and the roots die, it does not hold that really mucky organics in place. And as you have storm surge, it just literally is picking that material up and moving it offshore. So there's a lot of physical erosion that we're seeing as this transition occurs. Once you get through this short-term ghost forest transition, the peat starts building back up. The major problem there is, what's the next step in the evolution as you go from fresh to ghost forest to marsh is subaqueous. So this marsh will be submerged eventually with sea level rise. So where do we get beyond this as these subaerial environments are going underwater? Are we gaining or losing carbon or organic matter? Well, so we're starting to figure out what's going on subaerially. So this is a problem. We're losing organic matter, we're losing carbon. And if you're interested in global climate change and CO2 going in the atmosphere and all these types of things, this is problematic. And this is some initial data from our paired subaerial, subaqueous environments. So what happens when you go from your subaerial to subaqueous environment as the land is basically eroded and submerged underwater? So our initial data is showing significant loss. So this is an example from our mesohaline marsh sites. So we have 4 marsh sites that we have established, and this is just mean data. So it's the average thickness of peat on our y-axis here. And then along the X is distance. So think of this as a transect, and things to point out on this graph. 0 is shore. So this is a hard line. This is the current shoreline. As I mentioned in our methods, consistently we went to 20 meters in and did evaluations. So that's where we are here with our subaerial histosols. The average in the subaerial histosols of the marsh was 171 centimeters of peat. So we had solid typic Haplosapphoris or typic histosols, deep organics. This is the shoreline, and then everything to the right is subaqueous, so in blue. I have a little dashed line, and I'll show you what the significance of 75 meters is right now when we're talking about these marsh sites. Again, this is for 4 sites. We need more sites to really start thinking about mapping and carbon loss, but this is exciting information. So basically, I'm going to put a red line here at 40 centimeters thickness. So this is a magical line in soil taxonomy. When you have more than 40 centimeters of organic matter, you classify the soil as a histosol. When you have less than 40 centimeters of organic material, those soils are classified as mineral soils, typically entosols, inceptisols, altisols, whatever it is. This line is important in interacting with this line. At this point is where this gray line crosses this threshold. So what we're seeing here is in our subaerial environment, we have 171 centimeters of organics. Immediately offshore, once we go 4 or 5 meters offshore, our overall average thickness of peat goes from 171 to 78 centimeters of organics. It still classifies as a histosol, but it's a highly eroded wascyst. We've lost over 50% of the total carbon, or the total organics that were stored in that soil has been eroded and lost. So only 46 of the original amount of peat that was there is remaining, only like 4 meters offshore. This is problematic. Once we get to our offshore environments further off the shoreline, in this case for the marshes, it was an average of 147 meters that we moved for our offshore evaluations. There's 0 centimeters of organics remaining. So this line is representing basically the average depth of peat. And so what you're seeing is by the time you get to 75 meters offshore, you don't have histosols anymore. So these are all histosols over here. Once you get 75 meters and further off, these are entosols or wascents. So this is important for thinking about mapping. Where do we put map units in the subaqueous environment? So our wascysts or our eroded histosols still persist in the nearshore environment. In the cases of these marshes, that's within 75 meters of shore. So we have basically we have a histosol subaerial, and we have a histosol subaqueous right next to that. As you move offshore beyond 75 meters, we get our wascents. And this holds for all of our ecosystems. We're seeing this consistently over and over again. As you see the subaerial environment is being submerged, it is still maintaining the characteristics of a histosol, but basically we don't have any vegetation, no roots holding this in place, and wave action is beating up this organic matter. And once you have that site submerged for sufficient amount of time, that organic matter is going somewhere. It's completely lost from the soil system. So you basically go from 171 to 0 centimeters of organic horizons within 147, 150 meters of shore. This is problematic when you look at how fast our projections are of how fast these histosols are being submerged, 3.5 meters per year. So it's a lot of carbon that we're losing, and if you care about carbon accounting, this is a major problem. We're losing a lot of carbon very quickly as the landscapes are being submerged. Well, this brings up a major problem or philosophical question. We know the organic matter isn't in the soil system anymore, but where did it go? And we don't know yet. We need to do an actual carbon budget. So how much of that carbon from the soil system went into the water column as dissolved forms, as particulate forms, Did it move in the estuary? Did it move out of the estuary? How much of it went to the atmosphere? Right now, we have no clue on that, but what we do know from the soils perspective is we're going from 171 average centimeters in our marsh systems to 0 very quickly as submersion is occurring. And this is projected with sea level rise to increase and increase and increase over the next century or so. So our various things we need to test: is it eroding and moving in the estuary? Is it eroding and going to the ocean, a loss from the estuary? How much of that component is dissolved versus particulate? And then are the microbes able to mineralize it, which means turn that organic matter that's being churned up into CO2 or methane, which is a true loss from the terrestrial or aquatic system to the atmosphere. This is highly problematic for greenhouse gas production. Are we producing a lot of greenhouse gases as this submersion process is occurring in histosols? So further study is needed. We really need to look at these systems holistically, both our subaqueous and our subaerial environments. We need to really start understanding what's happening to our blue carbon. And the soil system is highly understudied in these overall systems, and we have a lot of work to do. Our estimates are about 3 million acres. About 2 million of that is in the subaqueous environment underwater, and about 1 million acres needs to be evaluated for advanced interpretations that are specific to coastal management and environmental interpretations. So our current soil survey information is not sufficient for that. This is the first project really getting us some information, getting our frameworks and moving forward. So the take-home message is we're having major shifts along the coast that are occurring currently. We're seeing massive changes in our ecosystems and soil systems as sea level rise and soil salinization is occurring. We really are working right at this point on baseline information. What is there right now? That is important because we have no clue. We need to know what our soils are like right now, and over the next 100 years, how are they projected to change, how that's going to affect interpretations for a number of things: agriculture, engineering, environmental interpretation, greenhouse gases, you can name them all. Subaqueous is massively important for interpretations like moorings, where to put docks, growing shellfish, aquaculture, all kinds of different interpretations we currently don't have related to soil survey in North Carolina. So we're a little behind certain areas of the country, but right now I think North Carolina is in a good place as far as getting the information and starting to think about how can we improve this for this information, the soil information for adaptation mitigation strategies for sea level rise. Along that sort of note, we have a number of additional projects that will be following these up. We're looking at geomorphic controls in 2021 of regional tidal marsh blue carbon. So we're moving out of the Albemarle Pamlico Sound and out towards the more salty Outer Banks systems. We're also going to start looking at our mineral soils as well as our histosols. So these pins here are our planned sites for looking at marshes. We're going to look at Swan Quarter National Wildlife Refuge. If you've ever been out there, it's miles and miles of marsh. And look at histosol catinas. When do we go from thick to thin organics, and where do we have mineral soils? Currituck marshes, we're going to look at our shallow organics, our Terek system. Out at Pea Island, we have the Carteret series, which is a high energy, sandy environment. These are entosols that are high in salinity. They're also very high in sulfur, very different system. And then down at Masonboro Island near Wilmington, we'll look at the Bohicket soil series, which is the most mapped salt marsh series on the East Coast. It exists from North Carolina down to Florida. It's a benchmark soil series. It's a very important soil series that we know very little about. And that's like highly fluidized clays that are high in sulfur. So there's a lot of interpretations that we need to be careful with dredging and things like that with the Bohicket soil series. And then something we're very excited about is we're moving away from the realm of wetlands and looking at, or wetlands and water, and we're looking at water, wetlands, and agricultural productivity in Hyde County, North Carolina, and looking specifically at soil salinization and the effects of salinization on farm fields in Hyde County. So that's sort of here highlighted in yellow, where we'll be looking at more agricultural interpretations. So remember, from the projections, this whole area is projected in the next century to be underwater. So we need to really start looking at these farm fields, thinking about salinization, thinking about soils. How can we help farmers and producers do their job with the number of aspects of sea level rise that are challenging and going to continue to be challenges into the future here in North Carolina. So with that, my ramblings are over. I really encourage you to check out some of the posters if you're interested in coastal zone soil survey. Really shout out to Ruben Wilson, our technician, did a lot of the work here, lots and lots of hours. Dean Shields with the NRCS, Rob and Debbie from NRCS, the whole coastal zone soil survey team through NRCS, the National Cooperative Soil Survey. This is funded through a cooperative agreement through USDA, NRCS. And if you're interested in sort of what's going on in pedology in North Carolina, I'm pretty active on Twitter. Going to be pretty busy this semester, but my handle is Muck Dragon, so pretty fitting that we're doing Histasol work. And here's some nice photos from the field, some Macaulay augers. Here's me in a tidal forest, and here's me in a tidal marsh. So I was at pretty much every one of these sites this summer. You know, really everyone, all hands on deck to get this done with COVID-19 restrictions. And so hopefully 2021 will be a little easier for us that work in the field. And, you know, obviously if you have any questions for me or anything like that, shoot me an email, or, you know, you can contact me on social media or something like that. So really appreciate the time. And with that, that's all for today. Thank you.